Okay, this is lecture 2 of chapter 42, the immune system, and this is going to be on specific immunity. Okay, the specific immune system has 4 kind of key components. One is specificity. So your specific immune system has to be specific to the antigen that it's attacking. The other is that it has to be able to distinguish self from non-self. So you don't want to be attacking your own cellular components or your own cells. You want to be able to recognize self from non-self. You also need to have a diversity. So if you're going to have this specificity and one particular receptor is going to bind to one particular molecule, you need a lot of those. And humans, we have about 10 million different receptors that can bind to different antigens. I can't get my head around that. That's too big of a number for me. And not only that, but then they have to find each other in your body, which I don't get, but it works. And then we have to have a memory so that if we get attacked again by that same thing, we can fight that off. So here they're showing you an antigen with the different epitopes, different binding sites, and the different antibodies that are specific to those. So you can see the shape has to match. Okay, there are 2 different types of responses, and understand that these 2 responses are working together, and they're working at the same time. So don't think of these as 2 separate processes. They're working together. So we have the humoral immune response, and the humors of the body are the fluids of the body. So B cells make antibodies, and these antibodies float around in the fluids of the body, and they're going to attack the different pathogen. And we'll have a whole lecture on how they go about doing that. The other type of response is the cellular immune response, and this is where cells become infected, and so the cell itself needs to get destroyed. And so this relies on cytotoxic T cells. So this is going to kill cells that have gotten bad, and these are going to attack things that are floating around in the fluids of your body. And so here again we have an antigen, and here we have a very simplistic model, but it has 3 different epitopes. And so these are the different epitopes. And an antigen, it's an antibody generating. So all this means is this is something that generates antibodies. That's where the name comes from. But your antibodies and your antigen have to have this specificity where they're going to bind. So you can see the shapes are important. Your antibodies have this Y shape, and the tips of them are where they bind. And if you notice here, the tips are going to be the exact same on an antibody, right? So this is going to have 2 of the same, 2 of the same. We'll see different shapes of the antibodies in the next lecture. Okay, the next is how we determine self from non-self. So this relies on these MHC proteins, or major histocompatibility complexes. They were named major histocompatibility complexes because they were found in doing skin grafting, and they didn't understand why skin grafting would sometimes be rejected, and that's where they identified these molecules. And so what these do is, here's an MHC1, class 1. These are found in all of your cells. This molecule will recognize self from non-self. So it will find antigens that are foreign, cradle them, and bring them to the surface of the cell. So it will not cradle your own molecules. It will only do this to molecules that are foreign. And so it presents these to the cytotoxic T cell. The other type of MHCs are only found on macrophages, or antigen-presenting cells, the cells that are going to engulf bacteria. And so this bacteria has been engulfed and broken down, and so a little fragment of it is picked up by this MHC, cradled and brought to the surface. And here a helper T cell will interact with it. Okay, and so kind of revisiting some signaling transduction pathways, which we've already done. So here we have an antigen-presenting cell, or a cell that's going to present antigen. And so this engulfs the bacteria, breaks it down. So remember, it brings it in a vesicle, which fuses with a lysosome, forming a secondary lysosome, which is going to then, as like a digestive sac, is going to break it down. And so then the MHC will cradle a piece of that foreign particle, bring it to the surface, and now this is going to bind specifically to a T helper cell that has a receptor that matches this particular antigen. There's another molecule here kind of for tight binding. I think of it as sort of as docking here, and this kind of holds it in place. And so some of the signaling transduction pathways, this activates interleukin-1 to be released. So this is a signaling molecule, which in turn causes the T helper cell to produce interleukin-2 and other signaling molecules. And these are positive feedback. So as you produce more of the interleukin, it activates the cell to produce even more interleukin. And then these signaling molecules then activate the cytotoxic T cell, which will attack infected cells, and the B cells, which will then be stimulated to produce antibodies. Okay, this is very similar, just going to have a different outcome. So here we have an infected cell, again, foreign particles. This case we have the MHC1s, and they're going to cradle the molecule, bring it to the surface, and that's going to activate a cytotoxic T cell. And again, we have a little tight binding here. This is CD8. And that's going to cause these perforin molecules to bind to the infected cell, causing a pore in the cell and causing that cell to lyse, killing the cell that's been infected. Okay, so what we're kind of describing here are both sides of the immune system. So the humoral immunity, again, is the fluids of the body. And the cellular immunity is when you have cells that need to be destroyed because they've been invaded. So here we have some kind of a cell with surface antigens. Here we have some free antigens, something that's going to cause antibodies to be produced. And so again, we have an antigen-presenting cell that's going to engulf these and break them down. And it's going to then present those with an MHC to the T helper cell. And the T helper cell will then in turn activate a B cell or activate a cytotoxic T cell. Cytotoxic T cells will then kill cells that have been infected that show the same antigen, because it will have receptors that match. And the B cells will secrete antibody, which can also bind because it's going to be specific to that particular antigen, or to the free antigen in this case. All right, we're going to end with this slide as well. Okay, so to look a little more specifically at the humoral response. So this part we've seen several times now. I'll go through it quickly. So again, antigen, some kind of antibody generating particle, gets engulfed by the macrophage. Macrophage engulfs it, breaks it down, presents it with the MHC2, activates a helper T cell. Remember, there's the interleukin 1, interleukin 2, which are signaling molecules. Once this T helper cell has been activated, and remember it has a receptor that's going to bind specifically to that particular antigen, it then causes the cloning of that particular cell. So we get lots and lots and lots of copies of those cells with those particular receptors. And then that T helper cells, now again, it's that amplification effect. So they're going to have lots and lots of these interacting with B cells. And so now this has to find a B cell that has a receptor that's going to bind to that specifically, and that's going to have antibodies that are going to match it. And so here we activate this B cell. That B cell in turn gets cloned. So we get lots and lots and lots of those B cells. Some of them become plasma cells. And in this case, you can kind of see the endoplasmic reticulum because it's making lots and lots and lots of proteins. These antibodies are proteins. So we're making lots and lots of these antibodies that match that original invader. We also get some memory cells, and those are going to be long lasting. So this just sort of gives you an idea. Here's a whole bunch of different B cells. Remember, you have like 10 million different types of B cells. And here's an antigen with its epitope. And so it activates, it matches this B cell. It's going to activate this B cell. So we get a cloning of this B cell. So we get this clonal selection. And then most of these are going to become plasma cells and secrete antibody. We'll have a whole lecture on how these antibodies work. And then some of them will produce memory cells, and those will stick around for subsequent invasions. Okay, on the other side of the table, we had the cellular immune response. So here again, we've done this a couple times. So we have this one's showing you probably a virus, and it's infected the cell. It gets broken down. Parts of this are presented with a class 1 MHC, activating a cytotoxic T cell. Once this cytotoxic T cell is activated, it becomes cloned. So you get this clonal selection of the T cell with the cellular receptor that matches that particular particle, and we get lots and lots of those. Those in turn will then cause those cells that have those MHC1s presenting it. It'll bind to those, and it'll then produce the perforin and destroy those cells. And so here's the cell being destroyed. This I think is kind of cool. This is the T cell, and you can see all the little surface cell receptors. And this is the cell that's been lysed. So here's the pore that's been produced in that cell to destroy that cell. Okay, and this is a last slide, and so you can kind of see how these two sides are interacting, right? So here we have our cell with the surface antigen and free antigens, and so we can have the T helper cells activating cytotoxic T cells to kill those. You can also have the B cells being produced, the antibodies, and we're going to see how the antibodies then can inactivate that or go after the free antigens. So I hope this helped. Our next lecture will be on antibodies.